Hello everyone, my name is Margarida and I'm a student from the second faculty. Today I will talk to you about the physiology of the nervous system. In this lecture we will cover homeostasis, the positive and negative feedback, cellular base of the nervous system, somatosensory system, physiology of pain, physiology of the olfactory, gustatory, auditory, vestibular, and finally physiology of the visual system. So, homeostasis. We can divide the word homeostasis into homeo and stasis. Homeo means similar and stasis means a condition. So, homeostasis is the state of maintaining a similar condition. To have a more general definition of homeostasis, we can say that homeostasis is the maintenance of a relatively constant internal environment. The variables that are regulated to maintain homeostasis include, for example, temperature, pH, the ion concentrations, the oxygen, the water, and failure to maintain homeostasis may result in disease. And if you look at the picture, we can divide disease into general groups according to their origin. And though we can divide in those that in which the problem arises from internal failure, and those that originate from an outside source. So for internal causes, we can mention, for example, the abnormal growth of cells, which cause the cancers and the benign tumors, the production of antibodies by the body against its own tissues, such as in autoimmune diseases, the premature death of cells and failure of cell process, such as in electrolyte disbalance. For external causes, we can mention, for example, toxic chemicals, physical trauma, foreign invaders, such as viruses and bacteria. To maintain the homeostasis, the human body, it monitors the condition, the functions I was mentioned before, that are the pH, the oxygen, the water, etc. And these functions, they should stay within a particular interval. So for example, the pH should be between 7.36 and 7.42, for example. The glucose levels, it should be kept between 70 and 100 milligrams per deciliter. The body temperature should be somewhere between 36.5 and 37.5. And the failure to maintain these values will lead to a disbalance that can cause the diseases. There are two basic patterns to control these mechanisms. We have the long-distance reflex control that, as the name says, it involves a long-distance pathway that uses the nervous system, the endocrine system, or even both. And the local control, that's the simplest homeostatic control that takes place at the tissue cell level. In the picture, you can see both systems represented, and you can see the difference between each of them. So for the local control, like I mentioned before, it's a simplest homeostatic control and is restricted to the tissue. And an example to understand this control is, for example, when we have a low oxygen concentration in a tissue, and this low concentration will be sensed by the small blood vessels, and they will start to release the nitric oxide. The nitric oxide will act locally, and it will cause the vasodilation, and which will increase the blood flow, and this increase of blood flow will bring more oxygen to the tissue. For the long-distance reflex control, we can divide this pathway into a response loop and a feedback loop, as you can see in the picture. The response loop begins when the stimulus is sensed by a sensor, and the sensor is linked by the input signal to the integrating center. And the integrated center, it decides on an appropriate response. The output signal travels from the integrating center to the target that carries out the appropriate response. Integrating centers are usually part of the nervous system or endocrine system, and the output signals, they may be chemical signals, electrical signals, or even a combination of both. And we can also say that there are two types of feedback loops, the negative feedback and the positive feedback that we will be talking now. In the negative feedback, the response opposes or removes the original stimulus, which in turn stops the response loop. The negative feedback loop can restore the normal state, but cannot prevent the initial disturbance, which is important to remember. And an example for a negative feedback loop is again the blood glucose levels. When the blood glucose level is too high, which is the stimulus, the pancreas will secrete insulin, and the insulin will decrease the blood glucose level, which is the response. In positive feedback loops, the response reinforces the stimulus rather than decreasing or removing it. And this might sound weird, but it will make sense with the example that I will give later. So, because the stimulus reinforces the loop, these will destabilize the system and initiate a vicious cycles of increasing response that sends the system out of control. This type of feedback requires some intervention or event from outside the loop to stop the response. One example of a positive feedback loop involves the hormonal control of the uterine contractions during childbirth. When the baby is ready to be delivered, it begins to put pressure on the cervix. The sensory signals from the cervix to the brain causes release of the hormone oxytocin, which causes the uterus to contract and push the baby even more against the cervix. And the increased stretch causes more oxytocin to release, which causes more contractions that push the baby harder against the cervix. So this is the vicious cycle that I was mentioned. And this cycle goes and goes until the baby is finally delivered, and this is the outside event of the loop that I will also mention. When the baby is delivered, these release the stretch of the cervix and stops the feedback loop. Now we will talk about the cells that we can find in the nervous system. So the basic cell, as you know, is the neurons, and the neurons are composed of a cell body with the nucleus and organelles that control the cellular activity. And then we have the dendrites that receive the incoming signals, and an axon that transmits the electrical signals from the cell body to the axon terminal. In the axon, we can find the nodes of Ravier that are sections of insulated membrane. The configuration of the cell body in relation to the dendrites of the axon can follow three basic patterns. So we have the pseudo-unipolar neurons that have a single process, which is the axon, that splits into two branches. One branch travels to the peripheral nervous system, and the other one to the central nervous system. They develop embryologically from the bipolar neurons, and this is why they are called pseudo-unipolar instead of unipolar. The bipolar neurons, they have a centrally placed cell body from which extend a single dendrite and a single axon. The bipolar neurons are common in the afferent pathways of the visual, auditory, and vestibular system that we will talk about later. The multipolar neurons, they are the most common type of neurons, and they possess an axon and a number of dendrites that arise directly from the cell body. The last one that I want to mention is the unipolar neurons that are not represented in the picture, but they possess a single process emerging from the cell body. And this type of neurons can be found in the dorsal root ganglia and sensory ganglia of the cranial nerves, and they constitute the primary afferents of the spinal cord. The last but not least are the interneurons, that are neurons that lie entirely in the CNS. In the nervous system, we can also find the glial cells. And the glial cells are supportive, non-neural cells that surround the neurons, the axons, and the dendrites. We can find different types of glial cells in the nervous system, in the central nervous system and the peripheral nervous system. So in the central nervous system, we can find 4 types of cells: the astrocytes, the oligodendrocytes, the microglia, and the ependymal cells. The astrocytes are the largest and most numerous cells in the gray matter. They form the blood-brain barrier, and they are also found in the limiting membrane that surrounds the brain and the spinal cord. They support metabolic changes, and they contribute to the energy metabolism of the central nervous system. And they also control the chemical environment around the neurons by clearing the neurotransmitters. Then we have the oligodendrocytes, and these ones, they surround and they myelinate axons in the central nervous system. And the ependymal cells, they line the ventricles in the brain and the central canal of the spinal cord. They are ciliated cells, and they are responsible to move the CSF through the central canal of the spinal cord. In the peripheral nervous system, we can find the Schwann cells and the satellite cells. The Schwann cells, they have a similar function as the oligodendrocytes in the CNS, so they are responsible to produce the myelin sheets that surrounds the axons. And the satellite cells, they are basically support the cell bodies. So now we'll start talking about the sensory system, and the best way to start is to understand the somatosensory pathways. The somatosensory system, it processes the information about the touch, the position, the temperature, and the pain. The receptors involved in transduction of these sensations are the mechanoreceptors that are responsible for touch and proprioception, the thermoreceptors that are responsible for temperature, and nociceptors that are responsible for pain and nauseous stimulus. There are 2 important pathways, as you can see in the picture, which are the dorsal column system and the anterolateral system. The dorsal column system processes the sensations of fine touch, pressure, 2-point discrimination, vibration, and proprioception. The primary afferent neurons have the cell bodies in the dorsal root, and their axons ascend ipsilateral to the nucleus gracilis and the nucleus cuneatus in the medulla. From the medulla, the second-order neuron cross the midline and ascend to the contralateral thalamus, where they synapse with the third-order neurons. The third-order neurons, they ascend to the somatosensory cortex, where they synapse with the fourth-order neuron. The anterolateral system is responsible to process the sensations of temperature, pain, and light touch. And in this one, it's a bit different. So the second-order neuron crosses the midline to the anterolateral quadrant of the spinal cord and ascends to the contralateral thalamus, where they synapse with the third-order neurons. And the third-order neurons, it ascends to the somatosensory cortex, where they synapse with the fourth-order neurons. In the picture, you can see the difference of these 2 systems. The somatosensory cortex can be represented by the homunculus, which maps the brain areas that are dedicated to the sensory processing for different anatomical divisions of the body. The largest areas, they represent the face, hands, and fingers, where the precise location is more important. So you can see that the size of the hands in the homunculus is much bigger than the size of the feet, for example. So because we have much more receptors in the hand than we have in the feet. The mechanical receptors, these receptors respond to many forms of physical contact, such as the stretch, pressure, movement, vibration, and texture. They are found in skin, but also in deeper regions of the body. And an important characteristic of these receptors is the type of adaptation that it exhibits. So we can have either very rapidly or slow adapting receptors. The very rapidly receptors, they detect changes in the stimulus, and therefore they also detect changes in the velocity. While the slow adapting receptors, they respond to the intensity and duration of the stimulus. And we have we can find different types of receptors in the skin, such as the Pacinian corpuscle, that are encapsulated receptors found deep in the dermis. They are the most rapidly adapting of all the mechanoreceptors, and because of their very rapid adaptation, they can detect changes in stimulus velocity and they code for the sensation of vibration. The Meissner corpuscle, they are also encapsulated receptors found in the dermis, most predominantly on the finger, lips, and other locations where the tactile discrimination is especially good. The Meissner corpuscles are rapidly adapting receptors that are used for the 2-point discrimination and precise location. The hair follicle receptors are nerve fibers surrounding the hair follicles. So when the hair in the skin is displaced, it excites the hair follicle receptors. And these receptors are also rapidly adapting and detect the velocity and direction of movement across the skin. The Ruffini corpuscles can be found in the dermis, but also in the joint capsules, for example, in the knee. And these receptors are stimulated when the skin is stretched and are slowly adapting receptors. The Merkel receptors and the tactile discs are also slow adapting receptors found in the superficial layers of the non-hairy skin. And these receptors detect the steady pressure and textures. Thermoreceptors are slow adapting receptors that detect changes in skin temperature. There are two classes of thermoreceptors, the cold receptors and the warm receptors. The cold receptors are sensitive to temperature lower than the body temperature, while the warm receptors are stimulated by temperatures that are higher than the body temperature. And when the skin is warmed above 37 degrees, for example, the cold receptors, they become inactive. And when the skin is cooled below the 37 degrees, the warm receptors become inactive. If skin temperature rises to damaging levels, so above 45 degrees, the warm receptors, they become inactive, and therefore warm receptors, they don't signal pain from the extreme heat. Instead, the nociceptors will be activated. So the nociceptors will be responsible to signal pain from the cerebral cortex. And the same happens with extremely cold temperatures that also activates nociceptors. The transduction of warm temperatures involve the receptors called transient receptor potential channels, TRP. And the fun fact about these receptors is that these channels can be activated by compounds present in spicy foods. And this phenomenon explains why people describe the taste of chili as hot. On the other hand, transduction of cold temperatures can be stimulated by compounds like menthol, which gives the cold sensation. So the pain. The pain is a subjective perception. It's the brain's interpretation of the sensory information transmitted along pathways that begin at the nociceptors. The receptors for pain are free nerve endings in the skin, muscle, and viscera. We can differentiate between fast and slow pain. The fast pain describes a sharp and localized and is rapidly transmitted to the central nervous system by the small and myelinated alpha-delta fibers, while the slow pain describes a duller and more diffuse and is carried on small and myelinated C fibers. The nociceptors are receptors that respond to a variety of strong nociceptive stimuli and can be caused by chemical, mechanical, thermal, and these stimuli they usually cause tissue damage. Important to remember that the nociceptors are not limited to the skin. but they can also found in visual organs. And we will talk about later about the referred pain. So the nociceptor activation is modulated by the local chemicals that they are released when there is tissue injury. So, for example, potassium, histamine, the prostaglandins released from the damaged cells, the serotonin released from platelets activated by the tissue damage, and substance P are all secreted by the tissues around where the tissue is being damaged, and these will activate the nociceptor. And the activation of these nociceptors will initiate a protective response, which is, for example, the reflexive withdrawal of the hand from a hot source, hot stove, for example. And this is a rapid unconsciousness protective reflex, and it's mediated by the spinal cord. So to summarize about the pain, we can say that the nociceptors, they detect a chemical, mechanical, or thermal nociceptive stimulus, and the conversion of the and this converts the stimulus to an electrical signal, which is the action potential. And then the C fibers and alpha delta fibers, they carry the afferent input to the dorsal horn of the spinal cord. And then we have the secondary where we have the secondary nociceptive neuron in the spinothalamic spinothalamic tract that carry afferent input to the thalamus in the CNS, and this will lead to the pain perception and the response that is set along the efferent pathway, and will result in the pain, the reaction to the pain. So as I mentioned before, we have the referred pain, which is the pain of visual origin that is refers to the sites of the skin and follows the dermatome walls. These sites are innervated by nerves that arise from the same segment of the spinal cord, which is, for example, if to give an example, in ischemic heart pain, the pain, you'll feel the pain in the chest, but also in the shoulder. The olfactory epithelium lies high within the nasal cavity, as you can see in the picture on the left. And on the picture on the right side, we can see that the olfactory nerve carries the information from the olfactory receptor cells to the olfactory bulb. And on the picture in the middle, we can see the olfactory epithelium. The olfactory epithelium consists of three types of cells: the supporting cells, the basal cells, and the olfactory receptor cells. The supporting cells are the pink long cells in the picture. The basal cells are the small triangular pink cells, and the olfactory receptor cells are the long blue cells with the cilia. The supporting cells are columnar epithelial cells lined with macrovilli at their mucosal border and filled with secretory granules. The basal cells are located at the base of the olfactory epithelium and are undifferentiated stem cells that continuously turn over and replace the olfactory receptor cells. These are the only neurons in the adult human that replace themselves, and it takes approximately 2 months to replace these cells. The olfactory receptor cells, which are also primary afferent neurons, are the site of odorant binding. The odorant molecule bind to the receptor on the cilia, which extend into the basal mucosa, nasal mucosa. The axon from the olfactory receptor cell leave the olfactory epithelium and travel to the olfactory bulb. These axons must pass through the cribriform place at the base of the skull to reach the olfactory bulb. So now a little bit more detail how the olfactory and the odorant olfact and the odorant molecules become action potential, and in the end how they reach the cerebral cortex. The odorant molecules, they bind to the specific olfactory receptor proteins located on the cilia of the olfactory receptor cells, as we saw in the pictures before. And the odorant receptor are G protein coupled membrane proteins, which in turn activates the adenylate cyclase, which will lead to an increase in the intracellular CMP. And these will open the sodium channels in the olfactory receptor membrane and produce a depolarizing receptor potential. The receptor potential depolarizes the initial segment of the axon, and action potential are generated and propagated along the olfactory nerve towards the olfactory bulb. The taste. So the taste is a combination of 5 sensations: the sweet, the sour, the salty, bitter, and umami. The taste receptors are located within the taste buds, on the tongue, palate, but also on the pharynx and larynx. The taste buds are anatomically similar to the olfactory epithelium and consist also in 3 types of cells. So the supporting cells, the basal cells, and receptor cells, as we can see in the picture. The supporting cells are found among the taste receptor cells, and important to mention that they do not respond to taste stimuli and their function is not known. The basal cells are undifferentiated stem cells that serve as a precursor to the taste receptor cells. And the taste receptor cells, which are the colorful cells on the picture, are the chemoreceptors of the taste system, and they line the taste buds. In contrast to the olfactory system, in which the receptor cells are the primary afferent neurons, in the gustatory system, the receptor cells are not neurons. They are specialized epithelial cells that function as the chemoreceptors, and they are responsible to transducing chemical stimuli into electrical signals. Each taste cells, they sense only one type of ligand. So each cell is responsible to detect the sour, the sweet, the salty, the bitter flavors. And these taste chemicals, they bind to the taste receptors on the microvilli, and they produce a depolarizing receptor potential in the receptor cell. And the ligands activate the taste cell, and there are various different intracellular pathways depending on which cell and which taste chemical. You can see it better on the picture on the left, that depending on which taste chemical binds to the specific cell, we will have like different pathways that will lead lately to the depolarization of the taste cell. And once we have this depolarization, we have an action potential that are sent to the brain. But how? So we have the cranial nerve 7, 9, and 10 that receive this information, and they take it to the brainstem. They ascend in the solitary tract, and they terminate on the second order taste neurons in the solitary nucleus. The second order neuron, they project mainly ipsilateral to the ventral posteromedial nucleus of the thalamus, and finally to the taste cortex, as we can see on the picture on the right. The auditory system. So hearing is our perception of the energy carried by the sound waves. The sound transduction turns air waves into mechanical vibration, and then to fluid waves, then to chemical signs, and finally to the action potential. We can anatomically, we can divide the ear into outer, middle, and inner ear. The outer ear is responsible to direct the sound waves into the auditory canal. In the middle ear, which is a hair-filled cavity that contains the tympanic membrane and the auditory ossicles, the malleolus, the incus, and the stapes. And the stapes inserts into the oval window, which is a membrane between the middle ear and the inner ear. The sound waves, they cause the tympanic membrane to vibrate. In turn, the ossicles vibrate also, pushing the stapes into the oval window, and the oval window will display a fluid in the inner ear. So the inner ear is the fluid-filled cavity that consists of the bone labyrinth, the semicircular canals, the cochlea and the vestibula, and a series of ducts that are called the membranous labyrinth. The cochlea of the inner ear contains the 3 parallel canals, and the cochlear canal contains the organ of Corti, which contains the hair cell receptors, the inner and outer hair cells, for the auditory stimuli. The cilia they protrude from the hair cells and are embedded in the tectorial membrane, as you can see in the picture on the bottom. The sound waves, they cause the vibration of the organ of Corti. And because the basilar membrane is more elastic than the tectorial membrane, the vibrations of the basilar membrane causes the hair cells to bend by a shearing force as they push against the tectorial membrane. And this leads to the bending of the cilia, which causes changes in the potassio-conductance of the hair cell membrane. And when the cilia bends in one direction, it causes depolarization, while bending in the opposite direction, it causes hyperpolarization. And the oscillating potential causes the intermittent firing of the cochlear nerve. And this is how the organ of Corti works. And causes the action potential that will be transmitted to the auditory pathway. So the fibers that ascend to the cochlear nerve, they go to the medulla, the pons, the midbrain, the thalamus, before they finish in the auditory cortex. And these fibers, they may be crossed or uncrossed, and as a result of this mixture of crossing and uncrossing to the ascending auditory fibers, to the ascending fibers, they represent both ears at a higher levels, and therefore the lesions of the cochlea in one ear causes unilateral deafness. But if we have a lesion that is closer to the brain, these won't cause unilateral deafness. The vestibular system is responsible for the equilibrium, and it detects angular and linear acceleration of the head. And it's responsible to adjustment of the reflex of the head and the eyes and the postural muscles, and these will provide a stable visual image and a steady posture. The vestibular organ is a membranous labyrinth consisting of 3 perpendicular semicircular canals, as you can see in the 2 pictures on the top. And the semicircular canals, they detect the angular acceleration or rotation, while the utricle and saccules, they detect the linear acceleration, as you can see on the picture C. And at the end of each canal, there is an enlarged chamber called the ampulla, which contains a sensory structure known as the crista. And the crista consists of hair cells and a gelatinous mass called the cupula. The hair cell cilia are embedded in this cupula that stretches from the floor to the ceiling of the ampulla, and it closes it. But how all these structures work in order to sense the rotation. So as the head turns, the membranous walls of the labyrinth, they move, but the fluid within the labyrinth cannot keep up because of the inertia, which is the tendency of a body to remain at rest. In the ampulla, the drag of endoleaf bends the cupula and its hair cells in the direction opposite to the direction in which the head is turning. If the stereocilia are bent towards the kinocilium, the hair cells depolarizes, causing the excitation. If the stereocilia are bent away from the kinocilium, the hair cell hyperpolarizes, leading to inhibition. Therefore, during the initial left rotation, the left horizontal canal is excited and the right horizontal canal is inhibited. And after several seconds, the endolymph catches up with the movement of the head, and the cilia returns to their original position and are no longer depolarized or hyperpolarized. If the head rotation stops suddenly, the fluid has built up a momentum and cannot stop immediately. And so the fluid continues rotate in the direction of the head rotation. And this is why people feel dizzy after doing several turns around the chair, for example. Finally, the last sensory system that we will talk about is the vision. So the vision is a translation of the reflected light into a mental image. There are photoreceptors in the retina that transduce the light energy into electrical signal that passes to the visual cortex where this image will be processed. The signals they pass through the bipolar neurons to the ganglion cells, whose axons form the optic nerve. And there is a special area in the retina. It's called the fovea, and as the most acute vision, because this is the smallest receptive field. The retina, it consists of 10 retinal layers, and the outermost layer is the retinal pigment epithelium, as you can see in the picture on the bottom right. And there is other 9 layers that are called the neural retina, that consists of 3 types of neural cells: the photoreceptor cells, which are the retinal rods and the cones, the bipolar cells, and the ganglion cells. The pigment epithelium, it contains melanin granules, and its function is to help in the formation of rhodopsin and storage of vitamin A, that we will talk about later, and to see how this is important to form these pictures. The pigment epithelium also provides nutrition to the photoreceptors, and it also absorbs light and prevents the reflection of the light. The rods and cones are the receptors, are the photoreceptor cells, and the rods are responsible for the monochromatic nighttime vision, while the cones are responsible for the high accurate vision and the color vision during the daytime. So the photosensitive element in vision is called rhodopsin, and is composed of opsin, which is a protein belonging to the superfamily of the G protein couple receptors, and retinal, which is an aldehyde formed from the vitamin A. So quickly summarizing, rhodopsin is composed of opsin, and retinal. The light on the retina converts the 11-cis rhodopsin to all-trans rhodopsin. And in this reaction, several intermediates are formed, and one of them is called metardopsin 2. The metardopsin 2 activates the G protein, and which in turn activates phosphodiesterase. And the phosphodiesterase catalyzes the conversion of cyclic guanosine monophosphate to 5-GMP, and cyclic GMP levels will decrease with this reaction. The decreased levels of cyclic GMP causes the closure of the sodium channels, which will decrease the inwards sodium current, and as a result, hypopolarization of the receptor cells membranes occur. And in this pathway, it's important to remember that the vitamin A is fundamental for the regeneration of 11-cis rhodopsin. And it's also important to remember, for example, that the lack of vitamin A, it causes night blindness. So after the light is converted to an action potential, it's transmitted to the cortex. And the axons of the ganglion cell from the optic nerve and the optic tract, that they end at the lateral geniculate body of the thalamus. And the fibers from each nasal hemiretina cross at the optic chiasma, as you can see in the picture. And the fibers from each temporal hemiretina, they remain ipsilateral. And therefore, fibers from the left nasal hemiretina and the fibers from the right temporal hemiretina, they form the right optic tract, and they synapse on the right lateral geniculate body. The fibers from the lateral geniculate body form the geniculocarine tract, and they pass to the occipital lobe of the cortex. So to understand this better is actually easy to understand if we imagine that there is cuts along this pathway. So if we cut the optic nerve, so it's the point A on the picture, it will cause blindness in the ipsilateral eye. If you make a cut in the optic chiasma, it causes the so-called heteronymous bitemporal heminioptia, which is represented by the letter B. And the cutting of the optic tract, so letter C, causes the homonymous contralateral heminiopia. And the last cut, that's right before the geniculocarine tract, causes the homonymous heminioptia with the macular sparing, as you can see in the picture D. So these are my sources, and hope this lecture was helpful. Thank you for your attention.